Welcome back to Hope in the Dose, the podcast series exploring the science, strategy, and collaboration behind bringing cell and gene therapies to patients who need them most. I'm Louise Kearney, Head of Cell and Gene Therapy, Strategy and Delivery at Fortrea, and I'll be your host for today's discussion. In this episode, we're focusing on a topic that's becoming increasingly central to cell and gene therapy development: external control arms. As many of these programs target pediatric or degenerative disease with small patient populations and limited or ineffective standard of care options, traditional randomized control arms aren't always feasible, ethical, or practical. External control arms can offer a powerful alternative, but they also raise important questions around study design, data quality, and regulatory expectations. To help us explore when and why external control arms may be appropriate, the different models available, and what regulators are looking for, I'm delighted to be joined by Christina Wassel, Director of Epidemiology at Fortrea. Hi, Christina. Thanks for joining us today.
No problem. It's great to be here.
So the first question I've got is, what are external control arms and why might they be needed for cell and gene therapy studies?
So external control arms basically refer to using data from other sources that are external to the trial as a comparator or control arm in a clinical trial. Especially for cell and gene therapy products and drugs, I think, these can be very effective ways to have a control or a comparator arm, without additional enrollment. Cell and gene therapy is a pretty unique class of drugs. So, standard control arms in clinical trials might not be super appropriate. They're they're just a very unique class and type of drug. In cell and gene therapy, these treatments are often for rare diseases, and often those diseases do not have any currently available treatments or effective treatments on the market, and a lot of times the clinical trials are pretty small. And I think one major consideration that to keep in mind when we think about external control arm for a cell and gene therapy trial is really to consider the likelihood that that type of trial design would be able to distinguish the effect of this cell and gene therapy drug from other factors that impact the outcome. And I think that that's pretty convincing for cell and gene therapy trials, because we would expect a large, almost curative effect in a lot of instances for these CGT drugs. And so I think that that's, one of the main considerations when you're thinking about an external control arm that, would make it a good choice, and why they may be needed.
Yeah, and we've certainly seen some results in the past where the results have been completely outstanding and different. So you can see why you could get away without an internal control arm or a placebo. Some of the studies that I've worked on in the past, they've been with children or they've been in degenerative diseases. And sometimes just the way that the cell and gene therapy can be quite burdensome to the patient as well, and difficult to set a placebo against. So I think some of those reasons are the ones that I've seen why clients have tried to go through the external control arm route.
Yes, agree. And those are really excellent. I think excellent examples as well.
So what are the options for external control arms?
So typically for external control arms, they're kind of separated into what we would consider a historical control, which would be a group of treated or untreated patients or participants from an earlier time period. That might be standard of care, might be completely untreated, whatever the standard of care might happen to be. That might not be much in a lot of these diseases. And the other option is kind of a concurrent group, which is basically a group of patients or participants treated during the same time period, but just outside of the trial in another setting. There's a lot of different data sources that can be used to create an external control arm. Data from other clinical trials can be used, electronic health records, claims, registries, and then other observational studies such as like natural history studies. And I think in particular, the natural history studies are of interest for CGT trials, because these, at least is my understanding, that the cell gene therapy trials often have, a natural history study preceding them. There are a few options for that in terms of patients from the natural history study who do not enroll in the trial either they don't consent or they don't have neutralizing antibodies. Those patients may go on to be served as an external control arm for the subsequent actual trial where patients are being treated. And then the other option is patients can be, serve as their own control, where they proceed through the natural history study where they're not treated, they're just followed, and then they enroll in the trial where they then are treated. And we just point out, I think the natural history study is of particular interest here. There are several other study designs and options that can be used. Each has their own pros and cons, that are specific to each type of ECA that you might want to choose.
Okay, thanks. I mean, I know that I've seen in the past with trials that I've worked on, especially in rare diseases, that the natural history arm, as well as being able to provide a control arm, can also take part of the education of the community within that disease area to learn a little bit more about clinical trials and in particular the gene therapy that might be coming up. And so they can also help as an aid to recruitment to the actual interventional trial as well. But there are pros, from what I'm hearing you're saying, there's pros and cons to different ways of doing things. So it's probably better to have those conversations with people with groups like Fortrea at the very beginning when you're looking at your whole program design to figure out what's going to work best for each particular circumstance.
Yeah, yes, absolutely agree. I think it's important to involve experts right at the beginning in this sort of study design, the design of the external control arms, both, experts and then also, the FDA, I think, which we'll talk about a little bit more, or other external, regulatory bodies, which I think we'll talk about in a few minutes.
And so what are the pros and cons of using an external control arm?
I'm just going to talk a little bit in general. A lot of these pros and cons I'm going to discuss really apply to what we would consider a lot of the observational, non-interventional space. So I think one of the things that is really always pointed out is bias. So there's several different types of bias, and really that goes for almost any observational study. Selection bias, information bias, confounding. That needs to be addressed in using an external control arm. There are a lot of ways to minimize bias through appropriate design, similar inclusion-exclusion criteria for the ECA and the trial, and then a lot of analytic and methodological techniques that, really can help to create a comparable external control arm. I think another thing that's kind of sometimes, people need to be sold on, or it needs to be, really addressed, is some data quality issues. Sometimes in observational studies, the stringency with which the data is collected is not quite the same as a trial. But, again, these can be mitigated if we know ahead of time that one of the purposes for, say, a natural history study is to serve as an ECA. We can upfront then really address that with excellent CRFs, data abstraction forms, whatever the case may be, really using stringent algorithms and definitions for a lot of the main, endpoints or main, exposures we want to look at. And, also using just good high quality data sources if we aren't in fact running our own observational study, if we're going to use a secondary data source, for example. So a couple of the kind of really good pros are cost and time efficiency. So, you don't need to enroll a standard of care or comparator control arm. This is already done. So it saves a lot, I think, in terms of patient enrollment. And I think, really is more efficient time-wise as well. I think the one thing that I think about is that then this means that all patients who enroll in the trial receive treatment. There's no randomization to some sort of standard of care or control group, which I think is a really nice feature, especially for cell and gene therapy.
Okay, thank you. And then just a little bit about any guidances. Are there any FDA guidances or guidances from other regulatory authorities that people should be aware of?
Yes, I think that, there are from the FDA, the most recent guidance is from February of 2023. It is still in draft guidance. It's not final guidance. at this point, but it's, really the one document for the FDA that we kind of have to go on. It covers design considerations for the external control arm, data source and data considerations, and then types of, analytic considerations as well, which really go, to what I was talking about before of, addressing those data quality and bias issues, maybe criticisms or critiques of using an external control arm. The FDA guidance that's out there now really focuses on the use of patient level data from other trials or other studies or real world data sources, such as registries, et cetera, and doesn't focus on things like aggregate level data, which can be used in some cases, but it's not the main focus of what's the guidance that's out now. Our advice would be to involve the FDA or any other regulatory authority early in discussions of external control arms, because you want to discuss with them the reasons you think that the ECA is warranted, your proposed data source, your analytic methods, your methods for addressing bias, your methods for ensuring that the ECA is in fact a true good comparator arm. And we at Fortrea do have a white paper and what we call our external control arm checklist available to assess the suitability of kind of using an ECA in different scenarios. And again, we at Fortrea can discuss, approach and strategy for using ECAs for FDA or other regulatory submissions and applications, and do have experience doing so.
Okay, so it sounds like there's a whole load of things to consider that perhaps people like me that are used to working in regular interventional trials might not be aware of. So it's a good idea to speak with someone with experience in order to work out the best plan for your molecule going forwards.
Yeah, because I think, really you do want to get the FDA or any other regulatory body on board early with your methods, with your proposal, with what you're considering doing, to make sure then that the rest of the, use of the ECA and the trial goes smoothly.
Christina, thank you so much for joining us today. This has been a really insightful conversation and a great reminder that while external control arms can offer significant advantages in cell and gene therapy, they require careful design, strong data foundations, and early engagement with regulators. For our listeners, the key takeaway is that external control arms aren't a one-size-fits-all solution, but when planned thoughtfully and supported by the right expertise, they can play a critical role in accelerating development while keeping patient needs front and centre. That's all for this episode of Hope in a Dose. Thank you for listening, and join us next time as we continue to explore the innovations and partnerships shaping the future of advanced therapies.